Alright, in this video we're going to talk about how we basically turn on and off genes, and we're also going to talk about the structure of chromosomes. What do chromosomes kind of look like? So when you take, right here, you have this word chromatin, and chromatin means the DNA plus the proteins that you kind of stick to DNA to give it the look of a chromosome. So the chromosome, DNA is never really by itself. There are proteins stuck all over it, and some of them we know of, like RNA polymerase, which can copy the DNA, but there are also proteins that stick on it to turn on and off those genes that we've talked about. And essentially, if something else sticks where the RNA polymerase should stick, well, then you basically have turned off that gene. And that's essentially what I'm going to try and explain to you here. But first, let's just talk about chromosomes in general. So here a picture of all of your 23 pair of chromosomes. So it only goes to chromosome 22. The last pair are the X and the Y. So chromosome 1 is the largest, and you get 2 of them. So here we go. We have 2 chromosomes. They are the longest. They have the most DNA, and you have 2 of them. And one of them, of course, you get from mom, and the other one you get from dad. All right, and then you go on to chromosome 2. It has a different set of genes on it, and yes, one of them you got from mom, and the other one you got from dad. And here we have them nice and pretty and lined up, and this is what they would look like. And chromosome 1 is always the largest, and chromosome 22 is the smallest. And then the 23rd set, they are not necessarily identical. If you have 2 X's, they are pretty much identical. I shouldn't say identical. I should say they have the same genes on them, and we'll talk about that in a different video. But essentially, all along here, there are genes, and we've talked about how the genes work and how you have a Tata box and a terminator, and all along here you're going to see these genes, and overall there are about 25,000 genes in all of these. But I want to talk about, we talked about in the last test, all these chromosome abnormalities. What happens when you have 2 or 3 X's, or what happens when you're missing one, or when you have only one X and you don't have a Y. Well, that was the one we called XO. And that just means you just had the X. Usually you can't survive with just one of your chromosomes. That is the exception to that rule, where you can have an X and no Y. The Y, of course, is what makes you male. If you have this, notice how much smaller it is than the X. There are fewer genes on it, but those are the genes that make you male. Over here, you have the X, and then if you have 2 X's, you're female. So this is a male here because it has an X and a Y, but if you have 2 X's, then you are female, and you have 2 copies of each of the genes on the X. So if you're male, you only have 1 copy of the X, but you have a few extra genes over here on the Y. And that's what makes you male, is the presence of the Y. Now you can be XY, then you have the wrong number, and we talked about that. That's called Klinefelters, I believe. I believe that was the name of the technical term, but it means you have 2 X's and a Y. So the idea here is this is what your chromosomes look like, and all along here there are genes. Now there's only a certain time that the chromosomes actually form. Most of the time inside the nucleus, these are all strung out and they're being copied. Right now they're all kind of curled up in the form of these chromosomes and getting ready to duplicate themselves. But otherwise you really don't see the chromosomes when you look in the nucleus, only at certain times. And we did a lab on that where you cut the onion and looked at it. And looked for the chromosomes. Now we're going to go inside the chromosome and kind of look to see what the parts look like. We'll also come back and get some terms that have to do with genetics in a different video. But right now we just want to know what makes up the chromosome. So here we have DNA, all right, and the DNA winds up, and then we get to these little things called histones, and histones are proteins that wrap around the DNA. All right, so here we have several histones here, and you can see the DNA wraps around it. And I've done a video where I kind of showed you that, maybe a little bit too complicated, so that's one reason I'm kind of doing this one. So once you have these histone proteins and you get DNA wrapped around it, you call it a nucleosome. So a nucleosome kind of wraps up the DNA. And then we put a whole bunch of these kind of wrapped up DNA in a row, and we call it the chromatin fiber. And then that wraps up into what we call a solenoid. And then that, of course, is what winds up into the DNA. So you can see that there's a lot of DNA in here. So the DNA wraps around these kind of wrap up, and then they coil up, and then that's what makes the little fiber that is bound back and forth to make up the chromosome. But there are pieces of DNA in between these nucleosomes, or in other words, there are little pieces of DNA that do not wrap around these histone proteins. And those are the open DNA. And we have open and closed DNA, and open DNA can get copied, and closed DNA, if you want to call it, they call it open and closed chromatin, but we'll just say DNA. And so DNA that is not wrapped around something else potentially can have RNA polymerase stick to it. So it's the little regions in between here, in between the little nucleosomes, those are the regions where the genes are on. The other genes are off. Now what we need to talk about, of course, is what turns them on and off. So let's move on to this next one. Here we're showing the DNA, and here we're showing it wrapping around the little histone proteins. Here it's showing you wrapping around the histones, and then it's showing it coiled up just like we saw, and then of course these go into the nucleus of the cell. So this is just another one. Don't worry about telomere or centromere. We're not going to talk much about that. But you do need to kind of get the idea of how this region in between, that's the key part right in here. In between those are going to be the open DNA, and those are going to be the places that the DNA is on. So what I'm trying to teach you now is what makes this region wrap around the histones, the histone proteins, and form a nucleosome, and what lets this piece not wrap around it and stay open so that RNA polymerase can stick to it and essentially leave that gene on. Well, it has to do with something like this. So as you look, here we have lots of room in between the histones. Here we don't have so much room. What made it wrap this way versus this way? That's what we want to know. We have condensed chromatin is what they'll say, an open chromatin. And the open chromatin, this region, there could be a TATA box in here somewhere. RNA polymerase could come in and attach and copy the DNA right here. Might not be able to do it right here, and certainly can't do it up here when it's wrapped like this. Actually, there are cases where it can happen. There are cases where you can move these around and shift the DNA on and off, but for the purpose of this video, we're just going to say it's either like this or it's like this. And here we get a lot more DNA, a lot more access to the DNA. Here we don't get much access to the DNA. And we need to know what it is that causes that. And the thing that causes it is something called methylation. All right, and I've mentioned it, but we're going to actually make a video here where we talk more about it. What happens is the DNA, wherever there is a C followed by a G, you have the potential to have what's called a methyl group. You don't have to know what a methyl group is. It's represented by these little lollipops, these yellow-orange lollipops here, kind of gold. And if they're added to a C that is next to a G, a C followed by a G, I should say, then that's what's going to make it stick to the histones. And you're going to end up with the closed chromatin like this versus the open chromatin like this. And of course, it's a very complicated process, but all we have to know is that this little chemical change that we make to the C's followed by G's is going to turn off genes. So if something comes along and causes these to be added or taken away, you are going to turn genes on and off. And if this of course relates back to Grandma's cookbook and our idea of cancer. Anything that can do this and doesn't do it right is going to cause cancer. And anything that does this or doesn't do it right might turn on and off a gene at the wrong time. For instance, hormones. You don't give hormones to little kids, Or, they'll start growing beards and stuff, and you don't want that. There's a story, if I haven't told it, I need to tell about hormones that where you don't want your kids being around them because, if they come in contact with, say, testosterone at too young of age, they'll start growing like beards and stuff. And that goes for little girls and little boys if they come in contact with it, because their cells are ready to absorb it. They haven't turned certain things off. So it's not just a case of turning genes on, but also turning them off. And you could end up with a beard, and you don't want a beard if you're a female usually. I guess if you want to join the circus, maybe. There's the bearded lady, and that would be a way to do it. All right, so understand that here we have the DNA methylated, and it would close the chromatin, and transcription is off. Now, this is not actually, this is a different type of methylation, but it works the same way. Essentially, you have closed off the DNA. Here the DNA is open, and therefore RNA polymerase right here can attach to a TATA and make your RNA and thus make your protein. If you're like this, isn't going to happen. All right, that's the key to this lecture. All right, now moving on forward, we need to now understand how this fits in with genetics. So we're doing genetics, and genetics is old school. It's the way we studied genes before we knew about DNA and certainly anything about methylation or nucleosomes or anything like that. We just studied it and we knew how it worked. We kind of located places on the chromosome where we knew that were important for producing genes, even though we didn't know exactly how they work. But we learned old school genetics so we can compare it with what you've learned in textbooks for years, high school, or if you're like my age, you learned it in college, 30 years ago. But that has changed, and I want to make sure that we understand that this is the new way that we understand things work. But you still have to learn the old way because the majority of the things you hear involve the terms, the old-time terms, which we're going to learn here in the next video I make, still apply, but we know the real truth about how everything works underneath the hood. It's kind of like describing a car without opening up the hood. Well, what I've done is I've explained how the engine works first, and now we're going to learn the terminology for, when you go to buy a car and they tell you it has certain features. Well, we don't know the name of those features, but we've already actually learned how they work. So I taught it a little backwards, but I think it's definitely the best way to do it from teaching this class several times. So make sure you watch this video first before you watch the one that I talk about where I call it terminology. The terminology one should come after this one.